Hi, everyone. Thank you very much for coming. It's my pleasure to get to tell you a little bit about the progress we've made in the past year and also give some information on kind of how we got to this point with our cell repository through ARC. Some of the progress that we've made has involved improving our efficiency using cryopreserved motor neurons and also improving our high content imaging capabilities and now expanding into TDP-43 related phenotypes. So you've heard a little bit about the ALS Research Collaborative, and Alan is going to give a much more extensive talk on the clinical side of this program. But for cell-based work, this program began in 2014, and at the time it was one of the earliest natural history studies of ALS that included the goal for collecting cell lines from patients with the purpose of reprogramming those into induced pluripotent stem cells. A lot of the early work for this program focused on building up this program, handling all of the samples that were collected from individuals, and then turning those cell lines into the induced pluripotent stem cells. This involved sending out DNA for whole genome sequencing, validating that these cells from patients or any donor doesn't have any pathogens, so they're safe to work with, and then taking the skin biopsy that was the initial collection material and generating fibroblast cell lines that could be used for cryopreservation and banking for use down the road, and then also for reprogramming those cells into induced pluripotent stem cells. I mentioned the majority of the lines early on were from fibroblasts, but in the past year, we've added the capability to the program to generate induced pluripotent stem cells from PBMCs, which are derived from blood. For all of the lines we generate, we do the requisite quality control assays to make sure that they are actually pluripotent stem cells. So this is a very simplified timeline of some of the early work that went into the ARC cell-based research program. As I mentioned at the beginning of the program, a lot of this effort was building up the program and collecting the cell lines that we can use today. Once enough of the cells had been generated, we kind of transitioned to starting some cellular phenotyping assays where we can look for disease phenotypes, developing our own models using genome editing of cell lines, and then finally trying to test therapeutic compounds on the cell lines and phenotypes that we've identified. So researchers were really excited with the advent of induced pluripotent stem cells because these cell lines could be used to differentiate into cell types that are affected in various diseases, especially with ALS, where you don't have generally access to nervous tissue. We use these cells from the induced pluripotent stem cells to make mainly spinal motor neurons, and then use them to perform what we call phenotypic assays, where we look for specific differences in the ALS cell lines compared to controls. If the phenotype is strong enough, we can attempt to use compound screens to look for small molecules that may rescue the phenotype, or also CRISPR gene editing based screens as well. And then the best performing compounds we can hand off to our pharmacology colleagues for testing in animals. At the beginning of this program, we mainly focused on SOD1 forms of ALS. This was due to the extensive knowledge at the time already of the role of SOD1 and some of the particular mutations, and also the in vivo pharmacology team's extensive experience testing drugs in mouse models of ALS. We also, around 2019, transitioned to adding C9orf72 modeling. This was due to the work by Fernando's team with Anna Gill and Alan Permasiri, where they showed that inhibition of type 1 PRMTs, or protein arginine methyltransferase enzymes, could reduce the toxicity of dipeptide repeat proteins. And these are proteins expressed by the C9orf expansion, and inhibiting type 1 PRMTs was beneficial. So we wanted to expand into C9orf72 biology so we could test if that hypothesis works in human neurons. And then more recently, we've transitioned to studying also TDP-43 biology, thanks to bringing on a talented researcher, Dr. Anushka Bhargava, who, in addition to studying cell lines from familial ALS patients, there were also, we brought in some commercially available genome edited lines from Jackson Labs in the INDY project, along with some genome edited in-house models that we made. So I mentioned we mainly work with spinal motor neurons, and you could ignore most of the details on this figure. This is our current preferred spinal motor neuron differentiation protocol. But the one thing I want to highlight is that it takes us 19 days generally to take a induced pluripotent stem cell on day 0 and do the requisite steps to turn those into young motor neurons. With those young motor neurons, then we can plate those for various assays that we're interested in, and then also at the same time do quality control experiments to make sure these are truly motor neurons and nothing went wrong during the differentiation, or there weren't any differences from cell line to cell line. Having to spend 19 days each time we wanted to do a differentiation meant that in order to have any sort of efficiency, we would have to overlap the differentiation with with the subsequent differentiation with the prior experiments from the first, kind of eating up a lot of our time. And so luckily, when we brought on Dr. TJ Kreistek and Anushka Bhargava, they had some suggestions for how we could modify our differentiation protocol. And that allowed us to attempt to cryopreserve or bank these neurons. So earlier this year, around February, we embarked on what was the largest differentiation we had collectively performed from 40 of the cell lines that we used the most. We differentiated them all to day 19 young motor neurons, and then harvested those cells and then froze them down in liquid nitrogen and performed batch quality control assays on all of the cell lines, giving us this bank of motor neurons from various forms of ALS that we're interested in. This was a dramatic improvement in our workflow because now we can just go to the freezer, take the cell lines that we want to study for a particular experiment, and then plate them and immediately restart at day 19, allowing us to improve our cycle time, which meant we could use our established assays more rapidly, but it also gave us the bandwidth to establish additional assays that we hope better model ALS. For today's talk, I won't be able to go too in-depth into some of the prior assays that we've developed because we've talked about that previously, and it's also, some of it's been published, but it spans looking at neuronal electrical activity to looking at survival of motor neurons when you apply an exogenous stressor. Here with oxidative stress, we've seen that SOD1 and C9orf mutant lines are more sensitive to hydrogen peroxide treatment. And then also C9orf lines are more sensitive to exogenous dipeptide repeat challenge. In the past year, we've enhanced our high content imaging capabilities in kind of two ways. One was by adding a automated confocal microscope, thanks to funding from Augie's Quest, and then more recently, a automated incubator that can supply plates to this new microscope 24/7 from funding from Mid-Atlantic ALS United. The second reason why we were able to kind of enhance our high content imaging capabilities was in the past year, we were able to recruit a highly talented neuroscientist, TJ, who's established the next three assays that I'm gonna discuss. So this is one of the first assays that TJ kind of established in our group based on some prior experience he had during his postdoc, and we call these spot cultures. So here you're looking at a highly concentrated cluster of motor neurons that are plated in a very low volume droplet on a dry well. This forces all of the neurons to attach in one small region. And because of this, you can then measure the axon growth from the spot and apply a type of analysis called Schall analysis, where you measure the number of axon intersections from increasingly further distance from the center of the spot. This is a robust assay in our hands. It's a lot easier to measure axon growth in this format than it would be in a dissociated culture, because axons would be kind of growing in all directions and it's hard to know which cells they're coming from. One of the issues early on, though, with this assay was plating manually was very time consuming, and it was also hard to accurately plate these cells in the same location across an entire plate, making it difficult to image these plates. So Amit on our team helped TJ fully automate this process, where now we can guarantee that the spot of motor neurons is in the same location in each well, allowing automated imaging of multiple plates. And this is just a representative panel of some of the data from cells that either have the C9orf expansion or where one or both copies was knocked out. So TJ applied this Schall analysis on a set of these neurons. So in blue here are 2 control lines that don't harbor the C9orf repeat expansion, compared to 2 lines that have the HRE expansion. What's important is this line was isogenically corrected from the line here. So this defect is specifically due to the presence of the expansion. You can see on the y-axis is the number of axon intersections. And then on the x-axis is further distance from the center of the sphere. And it appears there's much more robust axon growth in the control lines. Our team has been interested in the contributions of C9orf loss of function, gain of function mechanisms. So he applied the same type of analysis on cell lines where one or both copies of C9orf72 was genetically edited to delete expression. And we see that that also reduces axon growth in these cells. And you can apply area under the curve analysis to kind of simplify these readouts into a faster way to quickly look at the axon outgrowth in the different cell lines, where, again, it's confirming that with the C9 expansion, we're seeing dramatic reduction in growth. And without one or both copies of C9orf, there's also a reduction. So we're now starting to apply this assay to test therapeutic compounds. The second assay that TJ kind of helped scale up in our group was the ability to study axon regeneration. So for this, you plate all your neurons on one side of this microfluidic device, and then on the distal side, separated by microgrooves, you have higher concentrations of growth factors that coaxes the axons to grow towards the growth factors, and then you can isolate them. This allows us to then perform an axotomy or cutting of the axons. So here we're looking at an individual well where the axons were just severed. And then over time, we can image these same live cells and measure axon regeneration, plus or minus drugs, rescue compounds on different compartments. And so here is just a representative data set where we're treating with 7 different rescue compounds. For this panel, only one showed a significant increase in rescue above the DMSO control. And so that's a compound that we're going to continue to work with and pursue. The last high content imaging analysis assay that TJ has brought to us was the ability to measure lysosome transport. So lysosomes are organelles that are a part of the autophagy pathway. So this is a intracellular kind of recycling pathway where membranes, proteins, organelles can be recycled for reuse. And it's critical for all cells, but it's highly important for polarized neurons, where it can take days to transport content from the soma down to the axon terminals. And there's already been evidence from other groups that C9orf72 biology affects autophagy, either with gain of function mechanisms where the dipeptide repeat proteins can inhibit parts of this pathway, or loss of C9orf72 protein as well can alter the maturation of this pathway. So this is live cell imaging of either the HRE corrected, so these are wild type motor neurons, compared to motor neurons that have the C9orf expansion. And we're looking at cells that have been stained with the dye that accumulates in low pH organelles such as lysosomes, and measuring axonal transport in these neurites. This data can be kind of compressed by using these max projections, where any object that is moving generates a line, If it's stationary, it's just a dot. TJ can then apply particle analysis to more carefully analyze this. And some of that data is shown here, where the lines in the middle of these graphs are those are from the motor neurons that have the C9orf expansion. And there's a significant reduction in the percentage of mobile particles. Those that do move at a slower rate. They move less distance, along with a number of other parameters. We wanted to, again, look at the contribution of loss of function. So here are the motor neurons that have either one or both copies of C9orf72 deleted. And again, there's a reduction, although not as severe as in the HRE lines, in lysosome transport. And TJ's now begun to use this assay to test small molecules to see if we can improve this phenotype. So this is a representative data set where the red and green dots are the mutant lines, and then treating with different rescue compounds. Obviously, this one, rescue 2, made things a lot worse. So that's something we're not going to focus on. But rescue 3 had a significant increase in the transport parameters of these lysosomes in the mutant lines. And so we're following that up with trying to scale this assay up so we can do dose response curves. So transitioning a little bit from the high content imaging assays, in the past year and a half, we've added some models of TDP43 biology. And just as a quick bit of background, TDP43 is an RNA binding protein that's mainly localized in the nucleus, and it functions as a splice repressor. And TDP43 dysfunction leads to mislocalization from the nucleus to the cytoplasm, and then you lose that splice repression, so then you gain cryptic exons. So mRNA becomes dysregulated, and that's detrimental to cell survival. And we're interested in this because we hadn't been doing enough to study TDP43 in cells prior to this. And TDP43 pathology is estimated to be in up to 97% of all ALS patients. Interestingly, there's also data that links C9orf72 to TDP43 pathology. So here, a group expressed the C9 expansion in mice and saw that there was hyperphosphorylated TDP43 detected, or the expression of poly-GR dipeptide repeat led to TDP43 mislocalization out of the nucleus. So Anushka, with the help of Therese Dane, generated a TDP43 knockout panel so we can see what cells look like in the kind of most severe consequence when they fully lose TDP43. They lose Stavmin expression, one of the transcriptional targets, which has been shown previously. But surprisingly, these cells could still make spinal motor neurons, and they actually lived out to day 60 in culture. They appear to have altered neurite outgrowth. And now we're beginning to start to look into TDP43 mislocalization from the nucleus to the cytoplasm. So this is just a study where cells were treated with a translational inhibitor, puromycin. And you can see as you increase puromycin concentrations on the left or control lines, you get more mislocalization, but it appears to be worse in the cells that have the repeat expansion. So we're following this up with further assays. There's also been evidence that in cells with the C9orf repeat expansion, those motor neurons show signs of missplicing related to TDP43. So earlier this year, we performed a large motor neuron differentiation from several different cell lines and then aged them out to day 56. We treated with either type 1 PRMT inhibitors to see if that was a beneficial effect on this phenomenon, or also exogenous poly-GR to see if that worsened the situation. We collected RNA and then performed a nuclear and cytoplasmic fractionation. And then these samples were either sent out for RNA sequencing or in-house PCR, QRT-PCR assays to look for cryptic exon transcripts. So we're still analyzing the RNA-seq data that we're hoping to present at ALS MND later this year. But as a quick snapshot, some of our qPCR data from in-house experiments, we are detecting some cryptic exons in the C9 expanded lines. What's interesting is it's worse in the neuronal fraction of RNA, and that makes sense since splicing occurs in the nucleus. And so we're currently testing if PRMT inhibition is going to rescue this phenomenon that we see. So here I'm just summarizing some of the major phenotypes that we've observed in the different forms of familial ALS that we've studied so far. We are following up with various therapeutic strategies for each of these domains. And the findings that we've had in the years of studying these forms of familial ALS, we hope, and also the ability to generate cryopreserved motor neurons to gain more efficiency. We hope that in the coming few years, we're going to be able to kind of scale up this process and reuse it to start studying sporadic ALS, since that's a form that we haven't historically used with the cell lines that we have available. And in our cell bank through ARC, we have over 150 sporadic ALS cell lines. And then we can try to test these phenotypes that we've identified here in those models, and then also some of the therapeutic options as well. So I just wanted to thank our cell biology team. Yeah, I'm lucky to work with such an amazing group of people. And I didn't get to talk about all the projects that we work on day to day. So if you see them later today, please go up and ask what they're working on. And if you can't figure out what they look like from amidst 3D printed figures, this is actually what they look like. Also need to thank our past members who have generated the ARC cell bank, all the ARC participants, and then funding as well.